Okay, so let me start with the whole concept map here because I've been trying to record this several times. Had problems with video and audio. So the third macromolecule that you need to learn are your proteins. So your proteins are they are macromolecules, and they are made up of smaller monomers called amino acid. So amino acid attached to amino acid to attach to amino acid will create your protein macromolecule. So an amino acid structure looks somewhat like this. So it has a central carbon, and on one side it has an amino group, NH2, and on the opposite side you have the carboxylic group, which is the COOH, and here you have the H group, and on another side you have an R group. So it will have 4 different components on each side. Now the R group is called the variable group, and there are 20 different kinds of amino acid, each of which are made up of different R groups. So let me show you an example. Here you can see that the shaded areas right here, light blue, and then this is light brown, purple, and green. Those are the different R groups of the 20 different kinds of amino acids. Please notice this amino acid right here, glycine. It's made up of an amino group, a carboxyl group, hydrogen group, and the R group also happens to be a hydrogen. Okay, so don't get confused if this is used as part of your multiple choice on the test. Now, let's go back to our concept map. So if amino acids bind to another amino acid, they would form a dipeptide. So a dipeptide is a protein made up of 2 amino acids. If it's made up of about 20 amino acids, we call the protein an oligopeptide. And if it's made up of more than 20, hundreds to thousands of amino acids, these are really large protein molecules. We call it a polypeptide. Now, bonding of an amino acid to another amino acid also happens through the process of condensation. Well, let's look at how condensation occurs. Okay, so you have 2 amino acids here that will bind. What actually reacts are the amino of one amino acid and the carboxylic part of another amino acid. So remove OH from the carboxyl part of this amino acid, remove H from the amino part of the other amino acid, so H2O, that's going to be water molecule released. In the process, you're going to have a bond forming between the carbon of the carboxyl group of one amino acid to the nitrogen of the amino group of another amino acid. So you're going to form a peptide bond or peptide link between carbon and nitrogen, carboxyl and amino group of your amino acid. So the process of forming is called condensation, releasing water, and the opposite is hydrolysis also. So with the addition of water, we hydrolyze this dipeptide into its individual amino acid molecules. Okay, now you have, as mentioned, 20 different types of amino acids. Now, you only have 20 amino acids, but your amino acids can actually form very diverse, many kinds of proteins. Now, the magic number you have to remember is 20 raised to the power of n being the number of amino acids. So let's say, how many possible protein or how many possible sequences can you form if you have a dipeptide? So 20 raised to the second power, that's 20 times 20. Do the math. That's actually a simple math, but I can think right now. 20 times 20, that's about 400 different kinds of combination or sequences of your amino acid. Another example, if you have 4 amino acids, so how many structures can you form? So that's 20 times 20 times 20 times 20. That will give you 160,000 different sequences of amino acids. Now, when we talk about diversity, let's look at the shapes that your proteins can have. So either they are fibrous in their shape or globular. Fibers meaning the amino acids form elongated or linear structures or fibers. And globular, your amino acid sequences, the long sequences can start folding and form round or globular protein structures. Now, your proteins can also have different numbers of polypeptide. Just a quick review. Remember, a polypeptide is already made up of hundreds and thousands of amino acids. Now, some proteins are made up of more than one polypeptide. An example is your integrin. Integrin is a membrane protein that is integrated, hence the word integrin, integrated across your plasma membrane. It can serve as a channel where molecules can move in and out of your plasma membrane. So that is a protein made up of 2 polypeptides. A protein can also be made up of 3 polypeptides. An example is collagen, and these 3 polypeptides are intertwined, and collagen is actually fibrous in its shape. Another example is your hemoglobin. Your hemoglobin is made up of 4 polypeptides or 4 protein subunits, and your hemoglobin is actually globular in shape. So proteins are very diverse. They can take many different kinds of shapes, or they can be made up of different numbers of polypeptides. Now, all of these proteins that your cell is able to make and produce constitutes your proteome. Now, it kind of sounds like your genome. If your genome is all of the genes that your organism can produce, a proteome are all of the proteins that your cell can be able to produce. And we have a special technique of gel electrophoresis that you will learn, and this process can actually separate the different fragments of types of proteins that a cell can be able to produce. Okay, now memorization. You need to memorize, list and memorize the different functions of your protein. Sorry, let me rewind, backtrack a little bit. Again, I said the cell can make many kinds, many diverse shapes and structure of proteins. All of these shapes and structure has their corresponding function. You know, the shape is like this because the function is this. So many kinds of shapes and structure, therefore many different varied functions used by the cell. So the textbook lists many functions of different example proteins. Go ahead and memorize, let's say, at least 5 functions. So number 1, here you have catalysis. Enzymes are proteins. Enzymes are catalysts. Enzymes can increase the rate of reaction. Another example, contraction. So you have myosin, actin, or titin, protein molecules found in your muscle. So these protein molecules has their unique structure in which they can overlap and shorten the length of your muscle. Another protein is used for blood clotting. This protein is called fibrin, and fibrin is released in the process of blood clotting, and fibrin traps red blood cells and white blood cells in the area of injury so that it can increase the formation of a clot or a platelet plug. Another example are your hormones. So some hormones are made up of protein molecules, and you already know that hormones are needed by the body to signal and to balance the different metabolic processes. One more is immunity. So here we're talking about antibodies being produced by your white blood cells. So antibodies are protein molecules produced by your white blood cells to attack bacteria or viruses to help you from getting sick. Okay, so this is also new in your curriculum. You actually have to remember to explain from memory these 6 named proteins. So we have Rubisco. Rubisco is an enzyme that is created by your plant cells, and that enzyme is responsible in the light-independent stage of photosynthesis. Second one is your insulin. Your insulin is a hormone produced by your pancreas. So this hormone is a protein molecule that is released, produced by your pancreas, goes to your liver to signal your liver to take in glucose, so to help lower your blood sugar level. Third one is your immunoglobulin, or also known as your antibodies, which I just mentioned here. So these antibodies are protein molecules that combats bacteria and viruses. The fourth one is your rhodopsin. This protein is found in the rods in your eye. So you might be familiar with rods and cone cells. Those are specialized cells found in the retina that absorbs different wavelengths of light. Now your rod cells contains this protein called rhodopsin, and rhodopsin is used by your rod cells to be able to absorb specific wavelengths of light. Fifth, you have collagen. So collagen is normally found underneath your skin, in your connection between bones to bones, bones to muscles. And collagen is actually more structural in nature, so it gives a structure to different tissues. And then lastly, you have a spider silk. Now we're going outside of the body. We're looking at a protein produced by some organisms. So silk can actually be very strong, even stronger than I think it was steel, mentioned by the book. So we can utilize the strength from that protein produced by your spider and use that for other industrial purposes. So make sure you can elaborate and explain these named proteins. So what I want you to learn from here is that maybe two things. First, proteins are very diverse in their structure. Second, because they have diverse structure, well, it's just common sense to expect that there are also diverse functions or uses, purposes for your proteins. So make sure you can remember these for the test. All right, thank you.